Welcome to a fascinating journey into the cutting edge of science, where living bacteria and robotics merge to create something extraordinary. Today, we'll explore groundbreaking research from Carnegie Mellon University and the University of Minnesota that has developed microscopic robots powered not by batteries or electricity, but by colonies of living bacteria. These remarkable biohybrid machines represent a fundamental shift in how we think about robotics, bacterial intelligence, and the very boundaries between living and non-living systems. Join us as we dive into this microscopic world where science fiction becomes reality. At the intersection of synthetic biology and soft robotics, researchers have created a new class of robots that harness the power of living microorganisms. Led by Dr. Rachel Kalmar and Dr. Xinyu Liu, this research team published their groundbreaking findings in Science Robotics in February 2023. What makes these robots revolutionary is how they integrate living bacteria with engineered components, creating systems that can utilize biological capabilities that have evolved over billions of years. These include self-repair, energy efficiency, and the ability to respond to environmental changes, all while maintaining the designability aspects of traditional robotics. Unlike conventional machines that require external power sources and complex programming, these microscopic marvels operate using the natural movements and metabolic processes of bacteria themselves. The breakthrough involves a four-legged walker robot barely visible to the naked eye. This tiny machine consists of colonies of Bacillus subtilis bacteria encapsulated within a soft, flexible polymer matrix. The hydrogel matrix is composed of polyethylene glycol diacrylate, commonly known as PEGDA, which is infused with nutrient media. This specialized structure serves a dual purpose, providing both physical support and nourishment for the bacterial communities living inside. The fabrication process is incredibly precise, utilizing photolithography techniques to create microscale chambers within the hydrogel. These chambers are strategically positioned throughout the structure to maximize mechanical advantage when the bacteria exert force. When millions of rod-shaped B. subtilis cells align and move in coordinated patterns, they generate sufficient force, approximately 5 to 10 nanonewtons per cell, to deform the surrounding matrix and create movement. While the walking speed of about 5 micrometers per minute might seem slow, it's quite impressive considering the microscale dimensions of these robots and the fact that they operate without any external control system. What truly sets these biohybrid robots apart is how they leverage bacterial cognition and collective behavior. As Dr. Kalmar explains, we're not just using bacteria as tiny motors. We're leveraging their intrinsic sensing abilities and collective behaviors to create machines with emergent intelligence. Bacillus subtilis colonies exhibit sophisticated quorum sensing, a chemical communication system that allows individual cells to coordinate their behaviors based on population density. This communication happens through the release and detection of autoinducer molecules, which diffuse through the medium and trigger gene expression changes when they reach certain concentrations. Through this system, bacterial communities effectively solve complex navigational problems without any centralized control. When exposed to chemical gradients, temperature variations, or pH changes, the colonies adjust their movement patterns, steering the robot toward favorable conditions or away from harmful environments. Dr. Liu describes this phenomenon as a distributed biological computer, where bacteria perform parallel processing operations that would require significant computational resources in a traditional robot. This bacterial computation occurs through a complex interplay of gene regulatory networks, metabolic pathways, and mechanical interactions. For example, when nutrients become scarce in one region of the hydrogel, bacteria alter their flagella rotation patterns, creating differential forces that cause the robot to turn toward nutrient-rich areas. The research team has documented response times as short as 30 seconds for detection and reaction to environmental changes, remarkably fast for a biological system. Perhaps even more fascinating is the discovery that these bacterial communities can actually learn from repeated exposures to specific stimuli. The research has shown that the bacteria exhibit a primitive form of memory through epigenetic modifications and protein expression patterns that persist across bacterial generations within the robot. This means the bacterial colonies can adapt their responses based on past experiences, becoming more efficient at navigating or responding to familiar environmental conditions over time. This form of biological memory doesn't require the complex neural networks we associate with learning in higher organisms, yet it achieves similar adaptive results through completely different biological mechanisms. The bacteria essentially encode their experiences into their biochemistry, creating a form of distributed memory across the colony that influences future behavior. This emergent property of bacterial communities challenges our traditional understanding of learning and adaptation, suggesting that even simple single-celled organisms can exhibit forms of collective intelligence when organized in the right way. These microrobots, measuring less than 500 micrometers across, hold particular promise for revolutionary medical applications. The research team has already demonstrated that modified versions can navigate through simulated blood vessels and potentially deliver therapeutic payloads to specific tissues. By incorporating genetically engineered bacteria that secrete enzymes or therapeutic compounds in response to particular biomarkers, these robots could serve as programmable platforms for targeted drug delivery. Dr. Elena Vasquez, a collaborator at Johns Hopkins University, has begun testing variants designed to identify and respond to cancer-specific metabolites. The bacteria can detect chemical signatures at concentrations as low as 10 nanomolar, far more sensitive than many artificial sensors, Vasquez reports. And because they're living systems, they can amplify signals through their own metabolic processes. This extreme sensitivity makes these biohybrid robots potentially valuable for early disease detection, where they could identify biomarkers at concentrations too low for conventional diagnostic tools to detect. The biodegradable nature of these robots presents another significant advantage for medical applications. Unlike conventional medical devices that may need to be surgically removed or could leave behind synthetic materials, biohybrid systems can be designed to break down naturally after completing their tasks, leaving minimal traces in the body. Beyond medicine, these biohybrid designs show tremendous promise for environmental applications. Similar bacteria-powered robots could be deployed to detect and respond to pollutants in soil or water systems. A related study at Delft University of Technology has demonstrated that these robots can be engineered to target specific contaminants and either neutralize them directly or mark their location for subsequent remediation. Prototype systems have successfully identified arsenic contamination in groundwater samples at concentrations as low as 10 parts per billion, meeting or exceeding the sensitivity of many conventional detection methods. The biodegradable nature of these robots is particularly valuable in environmental contexts. When deployed in natural settings, they can perform their monitoring or remediation functions and then safely decompose without adding to environmental pollution. This represents a significant advantage over traditional environmental sensors or remediation technologies that often leave behind electronic waste or non-degradable components. These robots could be particularly useful in remote or difficult-to-access environments where deploying and retrieving conventional sensors would be challenging or disruptive. This technology raises profound philosophical questions about the boundaries between living and non-living systems. Dr. Sanjay Muralidhar, a bioethicist at Oxford University, points out that these creations occupy an ontological middle ground. These entities challenge our traditional categories, Muralidhar observes. They're neither fully artificial nor fully natural, but rather represent a new class of hybrid beings that blur the line between engineered and evolved systems. The work connects to emerging theories in cognitive science that propose intelligence as a fundamental property that can emerge at various levels of biological organization, from cellular communities to neural networks. Some philosophers of science suggest that these biohybrid systems may eventually help us reconceptualize intelligence itself, moving away from anthropocentric models toward a more inclusive understanding that recognizes the distributed and embodied nature of cognition across biological scales. This research challenges us to reconsider what constitutes life and intelligence. Are these properties limited to traditional organisms, or can they emerge in hybrid systems that combine biological and engineered components? The answers to these questions may have profound implications for how we understand ourselves and our relationship to both technology and other forms of life. As with many emerging technologies that blur traditional boundaries, regulatory frameworks have yet to fully address how such biohybrid technologies should be classified and governed. Are they organisms, devices, or something entirely new? Questions of containment, biosafety, and the ethics of creating partially living technologies remain active areas of discussion among scientists, ethicists, and policymakers. Current regulations typically separate living organisms from mechanical devices, but these biohybrid robots don't fit neatly into either category. This regulatory gap presents challenges for researchers and eventually for companies that might commercialize these technologies. Issues of containment are particularly important. How do we ensure that engineered bacterial systems don't escape into the environment or transfer genetic material to natural bacterial populations? Similarly, when used in medical applications, what safety protocols are needed to prevent unintended consequences within the human body? These questions highlight the need for interdisciplinary collaboration between scientists, ethicists, legal experts, and policymakers to develop appropriate governance frameworks for these revolutionary technologies. The research team is now exploring even more ambitious and complex architectures for their biohybrid robots. One particularly promising direction involves incorporating photosynthetic cyanobacteria that could power robots using only light energy, potentially enabling long-term autonomous operation. Preliminary experiments with Synechococcus elongatus have demonstrated that light-powered variants can operate continuously for up to 2 weeks without additional nutrient inputs. This represents a significant advancement in sustainability and operational longevity. By harnessing the natural process of photosynthesis, these robots could theoretically operate indefinitely in environments with sufficient light, converting solar energy directly into mechanical work. This approach eliminates the need for external power sources or nutrient replenishment, greatly expanding the potential applications for these technologies. The integration of photosynthetic bacteria also opens up possibilities for deploying these robots in remote outdoor environments where they could perform long-term monitoring or remediation tasks powered entirely by sunlight. These self-sustaining systems represent a fundamental shift in robotics design, moving away from energy-intensive traditional approaches toward biologically inspired systems that work in harmony with natural energy flows. Another frontier in this research involves creating multi-species bacterial communities within these robots, mimicking natural microbial consortia. Different bacterial species could perform complementary functions: sensing, locomotion, computation, and material synthesis, creating more sophisticated and resilient systems. Dr. Kalmer envisions a future where biohybrid robots might incorporate multiple microbial species working in symbiosis, similar to the complex ecosystems found in natural biofilms. "We're just beginning to understand how to design with biology rather than simply mimicking it," she explains. The potential for self-healing, adaptive materials that can respond intelligently to their environment is enormous. This approach draws inspiration from natural ecosystems where different species perform specialized roles while benefiting from their associations with others. For example, one bacterial species might specialize in sensing specific environmental conditions, while another might excel at generating movement, and a third could produce structural materials or therapeutic compounds. By combining these specialists in carefully designed arrangements, researchers could create robots with capabilities far beyond what any single bacterial species could provide. These multi-species systems might also exhibit greater robustness through redundancy and complementarity, similar to how natural ecosystems maintain stability through biodiversity. This research fundamentally challenges how we think about designing intelligent systems. As Dr. Kalmer concludes, what we're learning from these simple bacterial robots could fundamentally change how we think about designing intelligent systems. Sometimes the most sophisticated solutions don't require sophisticated components, just the right organization of simple elements working together. This insight represents a paradigm shift in robotics and artificial intelligence. Rather than building ever more complex individual components, the future may lie in organizing relatively simple elements into systems that exhibit emergent intelligence through their interactions. This approach mirrors how nature builds intelligence, not through centralized control but through the interactions of many simpler parts. From bacterial colonies to neural networks, complex behaviors emerge from the collective activities of relatively simple units. By embracing this design philosophy, engineers might create systems that are not only more capable but also more resilient, adaptable, and energy efficient than traditional approaches allow. As this technology develops, it represents not just an engineering achievement but a conceptual bridge between robotics, synthetic biology, and our understanding of collective intelligence in its most fundamental forms. As we've explored throughout this journey, bacterial robots represent far more than just a novel engineering achievement. They embody a fundamental reimagining of what technology can be. By merging living organisms with engineered structures, researchers have created systems that harness billions of years of evolutionary intelligence while maintaining human designability. These microscopic machines challenge our traditional boundaries between living and non-living, between evolved and engineered, and between individual and collective intelligence. From potential medical applications that could revolutionize drug delivery to environmental monitoring systems powered by sunlight, the practical implications are vast. Yet perhaps the most profound impact lies in how these technologies might transform our understanding of intelligence itself, revealing how complex behaviors and problem-solving abilities can emerge from the interactions of simple components organized in the right ways. As we stand at this frontier where biology and technology converge, bacterial robots invite us to reconsider not just how we build machines, but how we understand life, intelligence, and our relationship with the living world. The microscopic scale of these innovations belies their enormous implications for science, philosophy, and the future of human technology. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.